and just make a couple comments. You know, for me, one of the real nice things about being a professor is to mentor graduate students, you know, and kind of try to help them accomplish their goals of becoming, you know, critical scientists and contributing new information in a subject of interest. And when Mike joined my lab, he worked on a project on breeding multiple disease resistance in the dark red kidney beans. And the first project that he was assigned was trying to understand an unusual disease symptom phenotype that we were seeing on some of these varieties when they were deployed in the Midwest, mostly Wisconsin. And, you know, it was clearly a bacterial disease, but it didn't fit any of the known diseases, especially common bacterial blight that we were breeding for resistance. So Mike started on that, and as he was doing that and determining the etiology of that and developing into a good plant bacteriologist, he found his passion, and that was genomics. And he's done a really nice job on the genomics of xanthomonads that are associated with common bacterial blight. And that's kind of what you want to see. You want to see these, you know, a graduate student find their passion and then apply it and, you know, find and develop new information. So Mike's going to talk about that today. And he also served the department well, being a TA for Intro Plant Path with Rick Bostock and myself, Advanced Plant Pathology Lab, and the Plant Pathology module of the IPM course, PLS 105. So I'd like to introduce Mike, and before I step down, I want to thank Gita Coker, Dario Canto, Bill Lucas, who helped Mike with some editing of his final chapters while I was dealing with my medical situation. So thank you guys, okay? Really appreciate that.
So it's really nice to finally be up here. Oh, yeah. Great. So like Bob mentioned, I am a plant bacteriologist at this point, and I work on bacterial blights of bean. So what I'm going to do is talk a little bit about those, just generally, and then go into this more applied work, looking at halo blight and bacterial brown spot, and that unusual symptom that Bob mentioned. And then after that, we'll dig into the common bacterial blight genomics work that I did, and then wrap up with some of this genetic and mutational analysis of this interesting phenotype, brown pigment production, that one of the species that causes common bacterial blight are passed. So this is basic common bean bacterial blight crash course, right? This is halo blight caused by Pseudomonas syringae papiparphaseolipula. Bacterial brown spot caused by Pseudomonas syringae papipar syringae with some strains, and common bacterial blight caused by two different Xanthomonas species, Xanthomonas axanopodes papiparphaseoli and Xanthomonas fuscans subspecies fuscans. So just so you have an idea of what those look like in your mind. So halo blight is found in most bean growing regions, right? Principally affects leaves and pods. It is seed transmissible. It can infect them as well. Yield loss potential can be up to 45% under permissive conditions. It's currently divided into 9 races, the most economically relevant of which is race 6 because there's limited resistance to it. And it's generally favored by cooler temperatures. One interesting effect of this also is it makes a toxin called phaseolotoxin is responsible for this large chlorotic halo around these smaller necrotic lesions, right? It's also moves systemically, so you can get systemic chlorosis and some stunting of leaves and things like that. But the production of that can kind of taper off after like 23C, so it really is favored by this cooler temperature. Right? All right, bacterial brown spot. As you can see, it looks pretty similar in culture, right? Can't really tell it apart. It's found in temperate regions, primarily infects leaves. It can infect pods as well, but it's not nearly as severe as halo blight. There's less yield loss potential, up to 25% under permissive conditions. Unlike halo blight, it's not seed transmissible, but it can be found in association sometimes, not very common. There's no race structure on common wheat, right? And as you can see, symptoms are quite a bit different. You have these larger necrotic spots, they kind of expand or coalesce, center can fall out. There can be some chlorosis, but not too much, right? Not nearly as much as halo blight. So that brings us to this ambiguous symptom, right? So very clearly halo blight, very clearly bacterial brown spot. This one, what is that, right? It has some aspects of both. There's a lot of chlorosis, there's a lot of necrosis. It's hard to tell what it is. You can figure out by looking at this bacterial disease, you can see water and that kind of thing. And then you isolate and you get this Pseudomonas, right? Pretty consistently, but as I said, you can't tell. These are actually what these are. These aren't just the same plate. I promise you. I pulled them out of the freezer and took new pictures. Right? So these are actually what these are. So how did I approach figuring out what this is, right? So after tinkering around with this for a while, I settled onto this workflow, right? So there are some PCR primers that you can use to detect toxin genes that are used by the halo blight strains and bacterial brown spot strains, right? So one of them amplifies phaseolotoxin from halo blight strains, or at least a gene required for the biosynthesis of phaseolotoxin. The other one amplifies a gene required for the biosynthesis of syringamycin, right? But sometimes you can get these strains of halo blight that don't have that toxin. Very rare, but in Spain it's epidemiologically relevant. Syringamycin, it's not necessarily limited to bacterial brown spot strains. But the whole idea here is just to separate out strains that might be pathogen from other Pseudomonas that are growing on these, that can grow on the culture media or can be found in association with beans. Right? I'm just trying to reduce the amount of strains I actually have to deal with and narrow down what I want to find, right? Or narrow down what I'm looking for, right? The causal agent of this unusual symptom. So, so to that end, I also do a HR test for at least the suspected bacterial brown spot strains, right? To try to separate them from strains that are not going to be plant pathogenic, right? And then also take some strains and do a pathogenicity test just to confirm, right, that I have bacterial brown spot symptoms or halo blight symptoms versus something else, right? But again, this whole idea, instead of just jumping straight to a pathogenicity test and waiting the 10 to 14 days for symptoms to show up, so I can do the molecular assay, and then I have a pretty good idea of what I'm dealing with, and then just confirm that with the greenhouse assay. Okay, but what about those symptoms, right? That's what we really want to see. We don't, plant pathologists want to see sick plants. So I hope this is big enough that it's clear to you guys. So, so this is two of the cultivars that were out there, right? So this was a cultivar that had been grown in this region of Wisconsin for a long time, dark red kidney 85. And then this is the new cultivar, right, DRK 298, that these unusual symptoms were showing up on. So at this point, I should probably also mention most of the strains that I got out of, or I'd say all the strains that I got out of those unusual symptoms look like halo blight based on my assays, right? So take those strains, inoculate them on that newer cultivar. You see a lot of necrosis here versus the cultivar that was already out there. Very little, right? Still have that chlorotic halos around there, but here there's significantly more necrosis, right? Look at brown spot, looks about the same on both of them. There's not really much of a change here. Pretty typical brown spot symptoms of both of these. So, this led us into the conclusion this is probably an interaction of this particular cultivar with the halo blight strains that were already out there. I'm not going to talk about it on a slide for it, but I did a lot of DNA fingerprinting and that kind of stuff for the strains that were out there for halo blight, and they're all pretty uniform. So it doesn't look like it's something new. It just looks like it has to do with this particular cultivar, right? And it may be due, actually, surprisingly, based on what it looks like, to some resistance in one of the breeding parents. Because halo blight doesn't seem to cause severe losses on this cultivar later on. So, okay, is this applicable to strains from outside of Wisconsin? Yeah, this works pretty well. So, as part of my work, I get samples from different people that we work with or that know Bob and want to have a good diagnosis on some symptom that they're seeing. So in 2013, I got a bunch of samples of bacterial brown spot from Oregon and ran them through the same workflow. Worked really well. There's a really high correlation with that primer and the symptoms that we're seeing in the greenhouse on a top crop, which is a standard susceptible bean that makes these nice different halo blight and brown spot symptoms. We also worked with a group in Turkey, and they sent me a lot of samples starting in 2012. And again, it's the same thing. There's a really high correlation between what I'm seeing with the molecular results and then with the greenhouse results, right? So I can pretty accurately predict if I have a brown spot or a halo blight based on the molecular results. All right, so conclusions from this very applied section. These hybrid symptoms are probably, there's a cultivar and halo blight strain interaction, right? It doesn't seem like it's anything new, which is kind of too bad. It would have been a cool story, but that's what it is. The PCR primers that target phaseolitoxin and syringomycin biosynthesis genes can be multiplexed to differentiate halo blight and brown spot strains. That saves a lot of time. But in my opinion, the best practice for identifying these strains is still going to involve an HR or pathogenicity test, just to be sure, just because there's, it's possible that you can have these strains that can lose toxin genes, not that we've seen any, but they are out there somewhere in the world. So just want to be careful when you're doing the diagnosis, right? Okay. So moving on into the fun stuff, common bacterial blight genomics. I think it's fun. I heard somebody laugh. So common bacterial blight, this is a globally distributed seed transmissible disease of bean. It makes these really nice necrotic lesions with this yellow border, right? And these will expand as the disease progresses, right? It also makes these lesions on pods, so water-soaked spots. They can get kind of sunken or red, and they can lose bacteria, and conditions are good. This can cause extensive defoliation under permissive conditions, and yield losses can exceed 40%. So it's probably the most severe bacterial disease of bean out there. So it's caused by two different Xanthomonas species. As I mentioned, Xanthomonas accidenfolia. It's this guy right here, and Xanthomonas fuscans, subspecies fuscans. So these are not distinguishable when they're on beans, right? You can't tell them apart based on symptoms, right? But you can tell them apart in culture, mostly based on this brown pigment that fuscans produces, that I'll talk about a lot more in about 15 minutes. So there's no race structure on common bean, but there is pathogenic variability, right? So the Phaseoli strains tend to have, seem to have a host preference for Andean large-seeded gene pool beans. Fuscans have more of a preference for the Mesoamerican small-seeded beans, but you can also find them on Andean beans. And then, yeah, there is pathogenic variability between strains. And they really are distinct species. Fuscans used to be considered a variant of Phaseoli. No one wanted to elevate it to species level based on pigment production, or at least that didn't hold up. It keeps getting switched back and forth. But when you look at the molecular data, it really is a distinct species, right? So Fuscans is up here. These three strains are my representative Fuscans. These three are representative Phaseolis, right? So this is a multi-locus sequence alignment based on housekeeping genes. So yeah, distinct species within this Xanthomonas accidenfolia clade, which has a ton of pathobars that have been separated out into species, right? So one interesting case are these East African Phaseolis, right? So these are, they don't make the brown pigment in culture. Currently they're considered a Phaseoli strain, but based on molecular methods, clearly they look more like a fuscin, right? And one more thing, so sometimes you can get these non-pathogenic xanthomonads in association with bean debris or tissue, plants generally, right? And at least in the case of bean, we started wondering, okay, are these avirulent common bacterial blight strains, or are these an actual non-pathogenic xanthomonad that's also living in the same space, right? So I included one of these in my sequencing runs, and At least you can see it's a little bit out here, not really in the same group as Convector. But we'll talk about that in a little bit. Anyway, these are the 9 strains that I did whole genome sequencing for. So, you know a little bit more. How did I do that sequencing? Well, originally I tried Illumina, which is great for some purposes, but it wasn't so great for this, mostly because these genomes have a high number of repetitive elements in them that are longer than the read length for Illumina sequencing, right? So I can get draft genomes, no problem, but they're fragmented somewhere between 40 and 2,000 contigs. Not really what I want to be dealing with, right? Fortunately, and I have to thank Alex Greenspan for this, he said, oh, why don't you try PacBio? Because I was messing around with my Illumina reads at the same time we were TAing 120. And I looked at it and I went, oh man, you know, okay, so they're getting read lengths that are, at that time it was like, you know, 5 to 8 or 9 KB read lengths versus like 150 or 200 base pairs for Illumina, right? So I thought, okay, that could work really well for me because it will read right over these repetitive regions and it'll give me some spatial data on how these contigs are actually laid out, right? One of the other cool things, and this is, I think, pretty common now, but at the time, several years ago, I think it was a little bit less common, it was new to me, I was able to do all the assembly using Cloud Server. So I could run everything from my 8-year-old Dell laptop, right? It's not very powerful, but all I had to do was run the server. So that was really great. The drawbacks to PacBio generally, in case you're not too familiar with the sequencing stuff, so there is lower overall output. So this isn't great for every purpose, right? It's much more expensive per base than Illumina. In my case, since my genomes are small, I don't actually need that much output to get decent coverage, right? So this works well for me. Also, the error rate's pretty high, around 10%, but the error is random, so as long as you have sufficient coverage, you can wash out the error, right? Okay, so sequencing results. So this is pretty typical for Xanthomonas genomes. They're around 5 megabases, around 64 to 65% GC content. The coverage for my genomes ranges from 42 to almost 300x. It really just depends on how many cells that I need. I need 1 to 3 cells per strain, and part of that is where I actually got those really long reads and if I could reach those repetitive regions. There's 1 to 3 plasmids in these strains, except for, of course, the non-pathogenic strain, which is a bit different from the rest of the Convector live strains, unsurprisingly. Genome's a bit smaller, about 4.8 megabases instead of a little bit over 5. GC content's a little bit higher, right? Almost 66. And there's no plasmids in here. So that's interesting, right? So it starts to look like it's more of a non-pathogen versus one of these pathogenic ones. So this is a really big and colorful figure. It's a lot coming at you. If you haven't seen one of these before, this is a alignment using a program called MOB. So what this does is it takes the genomes and it tries to find what it calls locally collinear blocks. So these are regions of sequence where it's pretty much the same sequence in the same organization, right? So, so this blue block here, right, is the same as this blue block here, right? All these guys. So it's the same genes in there, same sequence, in about the same layout, right? So one of the reasons I call this one out specifically is one of the big differences between the Phaseoli strains and the Fuskin strains turn out to be this really big inversion around the replication origin, right? So about 0.7 megabases has been exchanged around that replication origin. You can also see that there's some variability in here within the species as well. So there's this big exchange here between these 2 Fuskin strains, right? And PX actually, this one of these Phaseoli strains, almost half or over of the genome is in reverse orientation compared to this Fuskin strain that I'm using as a reference, right? And the rest of these Phaseoli. So that probably doesn't have much of an impact on gene expression or anything like that because of the way these exchanges are done. But it's really really striking. So, and it illustrates these are, there's definitely some distance here between the species. The other thing that's worth mentioning here is that these East African strains have a much more similar layout to the Fuskin strains, right? They don't have this inversion that the globally predominant phaseoles have. Right? And then finally, there's these two blank areas here. So these are large insertions in the genome of these two strains, T21 and then that non-pathogenic strain, right? So it's interesting to look at a little bip more. So, especially because they're in the same location, right? But it turns out that they have different content. They do have a lot of hypothetical protein, type of reading frames, which is no surprise to anyone that's done genomics. There's numerous phage-related genes on both of these insertions, and some genes related to antibiotic resistance. Again, this isn't really too surprising considering the type of insertion. But the real striking features of this is for T21, that East African combacterial blight strain. There are genes related to type 6 secretion present. This is in addition to a type 6 secretion system that's already in the genome. There's also a type 3 effector, ZOP-E3. I'll talk a little bit more about the type 3 effectors in here in a minute. And then this non-pathogenic strain, most of it's taken up by these genes that are involved in non-ribosomal protein synthesis. Those kind of genes can be useful for competition with other microbes, right? So like in the biosphere. So that's probably what that's doing. All right. So as I mentioned before, there's a lot of mobile elements and repetitive sequence in these genomes, right? I think this is really cool. Other people need to. So the most striking thing about this is that East African strains actually have far fewer mobile elements than the phaseoli or Fuskin strains, right? So globally predominant phaseoles, around 130 to 150 copies of these different insertion sequences, right? Fuskin strains are between 100 and 160. These East African phaseoles, about 10 to 20 total copies. Significantly fewer, right? The non-pathogenic strain doesn't have any of these elements that are commonly associated with the common bacterial blight complex, right? The differences in what elements are actually in here break down along species lines for the most part. This element, this ISXAX1, is pretty common throughout the complex, right? But for example, this XFU2 has quite a few copies in Fuskin strains, but just a single one in the phaseoli strains, right? Again, this is reflecting some differences in life history of these species. And mobile element proliferation can also be associated with a increased selection pressure, sometimes with more virulent strains. And that comes back to this, right? So these strains are not necessarily as aggressive as the other phaseoles of the Fuskins, right? So this may be indicative that there's less selection pressure on these guys, or they're more of a progenitor strain, right? Okay, so let's talk about the plasmids, right? So there's 1 to 3 plasmids in each of these strains. They range in size from 1.9 kb to almost 60 kb, right? And they're all similar to previously described plasmids. And the non-pathogenic strain, again, lacks plasmids entirely, right? So, so this one here, this really tiny 1.9 kb plasmid, it's just in one strain, PX. This is really similar to a plasmid that's been described in a number of other xanthomonads. It has 2 open reading frames and no known function. So interesting that it's there. What it's doing, can't really say. These other 2 plasmids, PLA and PLC, are much more interesting. Right, so I'm naming them that based off of a previously published genome. That was this French Fuskin strain where they found 3 plasmids, which they named plasmid A, plasmid B, and plasmid C. It's PLA, PLB, C. Right? They're PLB plasmid, not present in any of my genomes, but PLA and PLC are. Right. So the PLA plasmid in my strains, it's present in all 8 of them, all the phaseoles, all the Fuskins. If you haven't spent much time looking at dot plots, this is a dot plot. What this does is it lays the sequence out along both these axes, right? If the sequence is the same at a position, there's a dot, right? So it makes this line if it's the same sequence. And then you can see some variability in regions like this where they don't share the same sequence, right? So if you look at in my Fuskin strains versus that previously published genome, it's pretty much the same plasmid, right? There's a little bit of variability, but not too much. You go over and you look in my Phaseoles, it's about the same thing. There's more variability, right? So this section not shared, but most of the plasmid is here, right? One thing that's interesting to note in here as well, so the Fuskin strains all carry a tau effector on this plasmid, as do the East African Phaseoles. But the rest of the Phaseoles, the predominant ones, do not, right? Okay, how about PLC, this other plasmid? So it's pretty much the same story, right? Fuskins, looks like it's almost essentially the same plasmid, a little bit of variability between strains. Phaseole, there are more differences, but most of the plasmid is still here, right? It's just some mobile elements and things like that have jumped in or out and changed the sequence a little bit, but the backbone of the plasmid is still there. This one is present in Fuskin strains and then the globally predominant Phaseoles, but not in the East African Phaseoles, right? And it carries a tau effector in all the strains of tau, as well as a homolog with other type 3 effectors, obviously. All right, so I keep mentioning type 3 secreted effectors. Really important virulence factors for gram-negative bacteria. Without a type 3 secretion system, Xanthomonas strains are virulent. So in my strains, there are 14 unique type 2 secreted effector families that are found in all of them, right? So this is probably a core, secreted effector repertoire, right? There's an additional 14 type 2 secreted effectors that are present in one or more of these strains, but not in all of them, right? So this typically breaks down along species lines. Again, Fuskins will have it. Globally predominant Phaseoles won't. There are 5 type 2 secreted effector families that are present, at least based on sequence, but they're pseudogenized in the strains that have them, right? So not functional. Why they're retained, I don't know, but it's interesting that they're there. Also, the non-pathogenic strain, as I mentioned before, it doesn't have those plasmids, right? It doesn't have type 3 secretion system, doesn't have type 3 secreted effectors. So it looks like it's more of a actual non-pathogenic xanthomonad versus a avirulent member of the combinatorial like species complex or some other pathway. So tau effectors in particular are really interesting. This was, I think, the first paper that I read when I started my PhD, which, if you know, when it came out, was a while ago. But Adam Bogdanov and Lubonis had just figured out the tau effector code, as it were, right? So tau effectors are DNA-binding proteins, and you can roughly predict what they're going to bind based on the sequence of the protein, right? They have a lot of specificity. And they function as transcriptional activators, right? So I was curious about tau effectors in my strains. So I predicted their DNA binding sites based on sequence of the protein. And at least for 2 of them, the plasmid-based ones, it's really conserved, right? It's actually the same in all the strains that have them, right? So the tau effector on plasmid PLA, same binding site in all the Fuskins and East African Phaseoles. For the tau effector on plasmid PLC, it's the same binding sequence on all the strains that have it. So same thing in the Phaseoles, globally predominant Phaseoles, and in the Fuskins, right? There are also several tau effectors on the chromosomes of globally predominant strains, but these all have different predicted binding sites, and then one of them, at least the PR1, is probably not functional due to a frame shift. Okay, so there's a lot of genomics coming at you real fast, so thank you for sticking with me. So the big thing here, at least based on aligning the genomes and looking at them out, is there's significant species-specific genomic rearrangements in these strains, right? It really reinforces this idea that Phaseolia and Fuskins are distinct species. So it's very nice when you get something that's consistent with all the literature that's out there. East African strains, though, are certainly distinct from the other Phaseolias, right? They look a lot more like Fuskins. So they might represent a progenitor population that gave rise to these Fuskin strains, right? So they're, in terms of phylogeny, they sit kind of basal to that clade. There's this hypothesis that Fuskin strains probably originated in Africa, so these may just be representative of that kind of population, right? There's some evidence for horizontal gene transfer among Phaseolia and Fuskin strains, especially with the plasmids. They're very similar, right? And then the non-pathogenic strain really seems to be a true non-pathogenic xanthomonad instead of just a virulent pathovar of infecting xanthomonad or something else, right? Okay, so let's get back to wet lab stuff before I lose some of you guys in the genomics. So as I mentioned, there's this really interesting brown pigment phenotype in Fuskin strains, right? So what is this? So this is a compound called iomelanin, right? It's an oxidized form of homogenistic acid. Somebody figured this out in the 90s, and then pretty much no one did anything with it. This isn't a really common characteristic of bacteria, but there are some bacteria that will make this compound. So at least in Fuskins, this is correlated with a loss of function in this gene, homogeneous state dehydrogenase, HMGA, right? So what happens is, the thought is that this gene gets knocked out through some mutation, right? Homogenistic acid accumulates, and it's about in culture media where it's oxidized and forms this brown pigment pile, right? So when I go and I look in my strains, this correlation holds. So all of the Fuskins have a frame shift in the same position, right? It's a mutation that makes HMGA probably not functional, right? Truncates the protein. Whereas the Phaseolia strains don't have this mutation, appears to be functional, right? So what I did, sorry, one more thing, try to So there's three things that I can test with this here, right? So I can knock out the gene HPPD, which makes homogenistic acid, right? And that should abolish pigment production. This is the only thing going on. The previous experiments determined that this was probably homogenistic acid were all biochemically based. They did it in culture, right? They didn't do any genetics. Another thing, knocking out HMGA in a non-pigmented Phaseolia strain should confer pigment production. This is the only thing involved, right? And expressing a functional copy of HMGA in a Fuskin strain should eliminate pigment production, or at least reduce it, right? It is complementing that non-functional gene. So I use this markerless deletion strategy, which is fun. It's a fun counter-selection thing. It's really, it's kind of a pain to get it working right, but when you get it, it's awesome. So I knocked out this gene HPPD, which again makes homogenistic acid in Fuskin strain, and look at that. There you go. Loses pigment production, right? So that was really cool to see. And then of course confirm that, and then there's no growth effect on this in culture, which is not terribly surprising because Fuskins have a mutation here, right, in this pathway already. Knocking out the gene upstream probably doesn't. Okay, but can I generate this phenotype in a non-pigmented Phaseolia strain, right? Yes, I can. I can delete the same strategy, HMGA, right? And I go from having this nice, non-pigmented Phaseolia to something that looks a lot like a Fuskin, right? Real nice. Again, it's not HMGA here, and there's no growth effect on this one in culture. And yeah, turns out I can also clone HMGA out of phaseoles, express it on a plasmid and control the neomycin promoter in a Fuskin strain, and eliminate pigment production there as well. So all this together really suggests that it's the loss of function of HMGA that's responsible for this Fuscus phenotype, right? It's brown pigment, biomelanin. All of the Fuskin strains have this mutation, right? Putting this gene, a functional version of this gene in Fuskins, it eliminates the brown pigment phenotype, right? Generating a similar mutation in phaseole, confers that phenotype. So that's pretty cool, right? But like, we really care about virulence effects because we're pharmacologists. So for some context, not always, but often, Fuskin strains are referred to as more aggressive or more pathogenic. It depends on the set of strains you're looking at, but there's some pretty aggressive Fuskins out there. And people will speculate in the literature for a long time, oh, maybe it has something to do with the pigment, right? The pigmented strains are more aggressive, so, right? Stands to reason. But, you know, that actually have mutants, no one was able to test it. I have mutants. So, that's the question, right? Is this related to myelin protection or other genetic factors? As I showed you in the previous section, there's quite a few genetic differences between these things, right? So I inoculated these wild-type and mutant strains onto 12 different bean cultivars that have different levels of resistance to common bacterial, right? So it's the susceptible, intermediate, and resistant varieties. But you know, it seems like there's no consistent, significant difference between these mutants and the wild-type strains, right? It doesn't seem like this has a direct impact on virulence, but that doesn't mean that it's not involved in you know, something else like survival, right? Biosphere, in some bacterial systems, some of the human pathogenic bacteria that make this similar compound. This is involved in like resistance to reactive oxygen species, at least in a culture environment. Right? So, and it could be something else going on there. Obviously, it's a fixed mutation in Fuskins, something going on there, but it doesn't seem to have a direct relation. Okay, so remember I mentioned earlier that I had a bunch of halo blight strains from Turkey, right? So one of the three of those strains make this suspicious-looking brown pigment in culture, right? It starts to look a bit like a Fuskin strain, and I think that's actually, I think you came in and looked and you said, oh, we can have Fuskins, and I said, no. You know, look again, so you see them, yeah, far away, right? So if you go and you dig back in the literature, it's not commonly reported, but some halo blight strains make a brown pigment that is referred to as similar to that produced by Xanthomonas Fuskins, subspecies Fuskins, right? So this is a cool opportunity to take a look and see what this actually was, right? Which is probably a mutation in the HMGA gene, right? Generating similar phenotype. So design primers amplified HMGA gene from these guys, the pigmented halo blight strains from Turkey, and from non-pigmented strains that I use as our lab strains. And also to this published genome of 1448A, kind of reference halo blight gene, right? And yeah, my pigmented strains have also a mutation in the HMGA gene, right? Again, it's loss of function in the same pathway. So it's not the same mutation, it's not the same position as in Fuskin, but it's in the same gene, right? So this is you know, a really interesting example of convergent evolution, right, between two pathogens of a common phenotype. And again, it's not a super common phenotype, but it is out there. It's nice to actually see that's what it is. So, all right, so conclusions from this section. So all the Fuskins strains that I analyzed have an identical loss of function mutation in the HMGA gene, right? Consistent with the literature, it's in the same position as every Fuskins that have a sequence for this gene, right? Loss of function of the HPPE gene eliminates pigment production. Again, that's the gene that synthesizes homogenistic acid, right? Provides genetic evidence that this Fuscous pigment is phenylalanine. HMGA strain, phasially strains with a HMGA deletion produce phenylalanine. It's consistent with the idea that loss of function of phenylalanine, or loss of function of HMGA results in phenylalanine production, and can complement Fuscin strains that produce pigment with a functional HMGA and eliminate or reduce phenylalanine production. However, gain or loss of this pigmented phenotype doesn't seem to impact virulence, at least in our experiments. And then some haloblite strains can produce phenylalanine through similar mutation as Fuscin strains, and that's really, I think that's a really interesting example of Persian evolution. There's got to be something there that's doing, that's going to be some benefit, but I don't know what it is. Anyway, acknowledgements. So I really like to thank Chippewa Valley Bean, especially Russell Doan and Bob Walksmith. So these guys, this company, this is the funding source. So these guys bankrolled my PhD, and I'm really happy, about that. Also, I got to thank my entire lab, especially Bob, who's been really great at pushing me to do a better job than I think I can do, and it's great. It turns out I actually, I can always do better than I think I can. I really appreciate that. And Maria has also been really helpful, especially with the greenhouse work with these mutants. Thank you so much. And Oscar and Lee Fung, if you guys remember them, they were really helpful in bounce ideas off of them a lot. Undergrad tech, Sandra helped a lot with making these mutants, right? Trees helped me a lot, thank you, with some of the genomics work. I got the vectors that I used from Pam's lab. Ferrung knew exactly what I was talking about, which was awesome, because no one else knew what a SACB gene was when I would ask them. UCDF Genome Center was great. Anna was really helpful. She made the libraries for me, and did these runs. Was super helpful at getting the PacBio working well for me. Nilesh, very helpful. I bounce ideas off of him all the time. Glad to see you made it. Alex, I'm not sure, is he a doctor now? I looked on the webpage and he was still listed as a grad student. But Alex gave me the idea again to look into PacBio or something. And then Lorena helped me a lot with like growth curves and that kind of work. So, and I'm sure there's other people that are not on here that I've forgotten, and I'm sorry that I forgot you. But everyone who's listened to me talk about this stuff over coffee or here has been super helpful at helping me think through my experiments. So yeah, and that's it. So we have a few minutes for questions, right?
I was wondering if for the halo blight pigment and non-pigment producer, do they have any different phenotype on plant, like when you infect beans?
Well, so I didn't see that. So I race typed those guys. They're both race 6. DNA fingerprints look the same, right? So it doesn't seem like they're actually different except for this mutation in that gene. I didn't do like side-by-side rating comparison or across different varieties. I just race typed them to make sure that I was still dealing with race 6, which is mostly what we were worried about when we were working with that group. So.
Why did you think that HMGA would be a player in
So generally, like once I knew that that's probably what this phenotype was, I didn't really think that this was probably going to be involved in virulence, but it's out there and it pops up often enough as a question that it's worth checking. The guy who was the first author on the paper that actually determined that this was pyromelanin actually came by to my poster at APS one year. It was like, oh, did you see a genetic basis for the pigment? Is it involved in virulence? Yeah, and that's, I mean, so it's worth checking. I was kind of skeptical that it would have an effect. It would be really cool if it did have an effect. And there still may be something from like an epidemiological standpoint, there could be a benefit, or from some kind of survival standpoint, there could be a benefit, but it's probably no direct effect. Is that an answer? Okay.
Did you do any biochemistry with pyomelon?
No.
Analytical,
No, I didn't.
Purified, maybe see if it had some toxicity?
No.
Sorry.
No. I know. I think that would be good. I mean, that's yeah. I mean, so I mean, you could probably purify it out of culture, right? So it makes it in culture.
Maybe Bob could keep you around for another.
If you help me with the biochemistry.
No, you showed us visual symptoms, maybe was like in the MGA meat. But did you do a bacterial titer on these?
No, I didn't do that. No. It's nice to hear him so popular, wasn't it? Yeah, that's probably something that would be more problem. Yeah. So we did 2 different rating points for this. We didn't do destructive sampling, but the other thing too is Phaseolia fuscans are different species. At least in culture, I can see if I do quantification, there can be different titers, right, based on the same need or something like that. So they may grow at different rates already, but I don't know if the mutants do or not. At least in culture, they don't seem to.
Do you think the pyomelanin might have some sort of role in relation to the environment that maybe you just weren't able to see in the particular environment that you were doing the experiment in?
Yeah, that's kind of what I think is probably going on. So like I mentioned, so this is what's really cool. I didn't know this stuff before I started writing my discussion section. So it turns out that a lot of other gram-negative bacteria do make pyomelanin also. Similar mutation, right? They lose the HMGA gene, they frame shift, truncate, something like that. So some Pseudomonas strains make this as a human opportunistic pathogen, Pseudomonas aeruginosa. Some strains have that, so at least in mouse systems, those strains are less virulent but more persistent. It's harder to clear the infection. Apparently Legionella strains make this, and there's some implication that helps them survive UV light. Some strains of Vibrio make this also. Same kind of thing. So I think there's something environmental, or there could be something about these particular hosts, but it probably has to do, I think, with survival of the environment, just based on that.
You did all this really amazing work about the biology of these organisms under the guise of diagnostics. If I were like an extension agent in Wisconsin needing to deal with a bunch of bean isolates, you had that great flow chart about diagnostics. Is there like a molecular test, or what advice do you have? What has this benefited, like a beginning diagnostician in bacterial bean diseases?
I feel like you have a couple questions in here, actually. But so, at least in terms of that workflow, the big takeaway for me is that there's a really strong correlation with these strains that are positive with Tyrannosaurus and bacterial brown spot. But also, I think it's still important to do those plant tests. And you really want to cover your butt when you're talking to somebody who's having to manage the disease problem, right? And you want to give them something as fast as you can, but you don't want to be wrong. So it's good to have an idea of what you're dealing with, but it's still good to check and make sure, right, by doing the plant-based assay, right? So, and
but you might want to
Yeah.
point out that in the end, those symptoms never became a problem.
Yeah.
And you trace that back to that resistance. So you'd be able to provide that information to a, extension person and say, really, on these varieties, this is not a significant disease, right? But, that type of information could go back to extension agents and, assure growers that this is not an economic problem, which that turned out to not be. So I have no questions. So what is the enzyme kind of complement of the pathogenic versus the non-pathogenic? Because those non-pathogenic ones are typically known to be tectolytic. So did you see a difference in terms of the enzymes? Because of course, xanthomonads make enzymes as well that may or may not be important in pathogenicity.
Yeah, so there are, let's see, it's been a while since I went actually looked at the enzymes. But there's some variability on in terms of what they have, but they all have like the cell wall breeding type enzymes and that kind of thing. There was one, and I tried making, I don't know if I tried making a mutant of one that was present in the common bacterial blight strain, but not in the non-pathogenic one, but I could never actually, it's a polygalactoronase, I could never actually get the mutant made. It didn't like the deletion. So the way that that deletion strategy should give me about 50-50 deletion mutants and wild type strains, and I couldn't get any deletion mutants for that one. So I don't know if it was a construct with that gene. I think that one's potentially interesting in terms of pathogenicity.
All right. Well, thank you, Mike, for a very nice seminar.